I'm here in two roles. One, I'll give a talk about my current hobby, but my other role is to write up what happened at this meeting. And one thing I'd like all of you to do is tell me what you're doing with Chemaxon, why you're at the meeting. Are you here just because you're interested in what Chemaxon is doing, or are you using it in some way that maybe even the company doesn't know about? So my email is there. If you don't see me during the meeting, please tell me. Okay, so yes, I am retired from Abbott, but they are kind enough to offer me a little office. There's room for me and nobody else, and hardly room for the 3 computers that are in there. So I serve as a consultant with them from time to time, but also do other kinds of things. Okay, so Tautomers have been an interest of mine for a long time, and it really popped up to the top of my attention about a year ago when Anthony Nichols did a symposium at the ACS, and he challenged us to say, what are the unsolved problems in drug design? And I thought, okay, Tautomers are really it. And so I gave sort of a small talk, and my interest has continued to grow to the point where now I've written an article, perspectives articles for Journal of Computer-Aided Molecular Design, and that spawned other things. But anybody interested in Tautomers, Terry Stout and I are hoping to organize a symposium at the fall ACS in Boston, so more or less a year from now, where anybody who's got anything to say about Tautomers is welcome. Okay, so why do I care about it? It's a challenge to every aspect of computer-aided drug design. First of all, Tautomers have different 2D and 3D structures. Okay, that affects a lot of things. But any kind of system in which we're measuring a property can have a mixture of different Tautomers. The major Tautomer depends on the environment, and there's no established way, no way that everyone agrees to predict the Tautomer ratio in various environments. Maybe water, but not much more. And so it affects everything from diversity selection, because obviously if you have the wrong structure in the computer, you're going to think this molecule is different from another one when in fact it really isn't. Calculating physical properties. Certainly the big issue is with docking and scoring, but also because the structures of the Tautomers are different, clustering might put 2 molecules that really are the same in different clusters. And then, of course, there's the issue of quantitating the effect of tautomerization on biological potency. So you say, oh, well, that's not such a big problem. But in fact, it is. These are marketed drugs, so they're generally smaller than the stuff that we're working on these days. But 25% of the molecules of marketed drugs have more than 1 Tautomer. And you see way down at the end, there are many that have more than 10. Okay, so just now, I'm sure there are people here who aren't chemists and wonder, what is a Tautomer? So I have to show you some structures to get you on the page. So this is a famous one. 2-aminopyridine exists as the aromatic form, whereas substituting the amino with an OH group, you go to the pyridone form. And this little arrow here is to point out that the equilibrium constant has a negative sign. And same way, the thio. I should point out, Alan Katritsky is one of the organic chemists who's worried about this a lot. But it gets more complicated than that, because here again I have the 4-hydroxypyridine in the pyridone form, but if you add 2 chloros, all of a sudden it prefers the hydroxy form. And if you put it in this heterocycle, again it prefers the hydroxyl form. So this says you can't just sort of write some rules and expect that you're going to predict the tautomer form. It's more subtle than that, which is why we're in chemistry. Here's a tautomer equilibrium that's not so commonly thought of those who write tautomer programs, and that is ring chain. And in DMSO, this particular cyclization, it's an equilibrium. It's a little bit slow, but you see it changes from being equal if there's no substitution at the 2 position, sorry. But if you can change the ratio by changing the substituent. And this paper has a lot more compounds, but they showed it follows a Hammett relationship. So, it should, but you're going to do a Hammett relationship for every kind of tautomer? Okay, so the challenge for chemoinformatics is a compound can have more than one structure. So one way to solve that problem is to store a unique tautomer and say, well, that represents the whole family. And that will help you get you a certain distance. But let me show you a problem. Okay, so here are 2 very closely related compounds. They differ by a methyl group. Let's pretend that you're a laboratory person and you've been measuring partition coefficients and been careful to figure out what tautomer is in what solvent. And you want to store that so that as you do this every day for many, many compounds, you ultimately can develop rules to predict tautomer ratio. Okay, so for each compound, there are 3 structures, and there are 2 solvents. And so how would we keep this? We almost need a relational database between the unique tautomer and then all of these specific ones, and then associated with each one the fraction in that particular solvent, or somehow keep the tautomer ratio, so the ratio of those 2 in a particular solvent. So this is not a solved problem. I've not seen anyone who can do that. Now, to be fair, there isn't a lot of this kind of data. Some of it exists, and part of the whole problem of tautomerization is that there isn't a lot of this data because it's darn hard to measure these things. Sometimes the tautomers don't have different physical properties, so you don't even know what you have. But related to this, then, is people who measure, say, pKa's or Octanol-water partition coefficients, our favorite QSAR properties, and they write down a structure of a molecule that they did, but they're different. You can see here there are different tautomers in different phases, and that's probably not recorded. So that affects then the programs that we use to calculate those kinds of physical properties. And if we can't calculate log P, then all of our triage of rule of 5 and all that stuff, well, that's a little suspect also. And I'll just show you an example. So Viagra has 3 tautomers. Maybe there are more, but 3 that I did. Interestingly, in PubChem, it's shown as 1 tautomer, but Chemical Abstracts has a different tautomer, and this is the one that the original people published, but who knows what it is when it's acting. But now look at 3 of the accepted Octanol-water partition coefficient programs. C log P and A log P say that the hydroxy tautomer is the more hydrophobic, but Cowan says no, it's this particular tautomer, or the hydroxy is the least hydrophobic. So this is a problem for the young people in the audience to be thinking about. I can tell you what the problems are, but I don't have a solution. Okay, and so just to remind you, 4-aminopyridine exists in this form. It's not very similar with the cytigic fingerprints, so unsimilar that there's less than 1% chance that we would say those should have equal potency. If you're looking for similarity to find other molecules that might be active, you might pull this out and say, oh, well, that has a 19% chance of have similar potency. But unfortunately, it's the minor tautomer. And so you're stuck. Now, I just want to show you some observations from high-resolution crystal structures. I'm pretty sure there are more of these cases, but the crystallographers often, they just slap a small molecule in there, and their force fields aren't so good for small molecules. And so some of even the high-resolution structures, less than 1 angstrom, probably need to be re-refined. But some have been published, and this is the favorite one of everybody. It was published in 2001, where this barbiturate, which this is the only structure that most people would say is a stable form of it. But in fact, the enzyme wants to move the protons around, move this proton to the OH group in order to find the binding. And probably because it's a metal, the metals pull over here somewhere. Here's an interesting one. There's very little difference between the two tautomers. The hydrogen either here or here. AM1 calculations say this one is preferred by 4 kilocalories, and yet the enzyme binds that. Carbonic anhydrase actually binds this, instead of the amide form of this molecule, the hydroxyamine. This is a really cute example. The inhibitor of xylose isomerase is xylitol, and it's bound. There's a nice crystal structure from Ringa and Petsko. And so if you were modeling and say, oh, I want another inhibitor, you'd fool around and change the OH groups to chloros or whatever. But in fact, the substrate, they can capture the substrate. The substrate is glucose, and it binds in a ring form, not in the open chain form. So these enzymes are really clever. This is from Enzyme Kinetics, MIF, 2 tautomers. Now those, because it's moving hydrogen to a carbon, that equilibration is slow, and so they can actually isolate both tautomers and put them in and measure the kinetic constants. And so the enzyme recognizes both of them, turns them over both equally well, but recognizes the enol form 10 times better. And again, you see the Octanol-water partition coefficients don't agree with the 2 solvents, 2 compounds. CDK recognizes both forms. Now this is a really cute structure. It's very high resolution, and you can see in the structure the two tautomers bound. However, 2 other kinases, KDR and PDF, only this tautomer is bound. Okay. And then, of course, there's the case when a molecule can have lots of tautomers. And what do you do? Okay, tetracycline. If you were trying to figure out what is the biologically active form of this, or which ones probably, there are 19 cationic, so putting proton here, and neutral tautomers. This 8-oxoguanine is a mutagen, and there's one paper devoted to it. It has 100 neutral or anionic tautomers. And so how are you going to figure out what it might be? Now, you might have some analogs that'll help you, but still, the baseline is this is a mutagen. You'd like to know how it's doing its job. Okay, and so my long-term interest, of course, has been quantitative structure-activity relationships. And so how do you handle it, especially if you have a series where some of the molecules can tautomerize and others can't? How do you penalize that some of the molecule is wasted by being in the wrong form? And related to that, is there a realistic cutoff in the tautomer fraction that can be used to eliminate structures for virtual screening? And you saw from these protein examples that some of those that barbiturate was calculated to be 20 kilocalories higher in energy than in water, than the low energy form. And so what are you going to do? So the bottom line is tautomerization remains a challenge to 2D and 3D QSAR, docking and scoring, diversity and similarity, filtering and data mining based on physical properties. And so I thank you for your attention and welcome comments, arrows, whatever. Thank you very much.
We have time for a few questions, so starting with...
Hi.
Hi, I'm Steve Boyer
Yes.
With IBM Research, and I'm curious to know if you are aware of situations where a company, for instance, has patented one particular compound, and they actually get a patent and they draw the structure as, say, A, and then along comes another company, B, and they take a tautomer, which they isolate and crystallize, and say, well, you know what, we have the structure B, and that structure was not defined in the patent. And are you aware of any kind of situations like that?
No, I would defer to you. You're the patent expert. Start looking for them.
Actually, I am. It was 3-hydroxycephalosporin derivatives, but where the keto and the enol form, and finally some of the guys actually crystallized the keto form and took an X-ray. And it gets into some very interesting legal questions, I guess, when it comes down to the intellectual property issues.
Yes.
So that's all. I'm just curious if you had any examples.
Hi, Yvonne. Pat Walters, Vertex Pharmaceuticals. So, as someone who spends entirely too much time thinking about virtual screening, the thing that concerns me about Tautomers is we all know that the thing that kills us in virtual screening is false positives. And I think if you start enumerating out too many Tautomers, and you're not really, really careful about this, you're going to actually end up making your life more difficult by getting more false positives with perhaps unrealistic Tautomers. What do you think about that idea?
I guess I would say, certainly for docking, right, that's a problem.
Yeah.
But then what I would hope someday we'd be able to figure out is to do a good scoring or a good optimization and say, oh, well, yeah, this thing can bind, but you know, here are all of the energetics and so on. And so, you know, I don't know the way forward for calculating these binding affinity, if we have to do QMMM or if something in between will come. But I would say just docking, right, part of the thing is if you dock and you say, well, I'll take the 1,000 best, you know, what's your objective? Do you want to find everything? Well, you'll never find everything unless you screen the whole library. Do you want to find most of them, enrichment, and so on? Some people have shown that adding more Tautomers, you do get a little better recovery, but it's a big issue. And think about it. Tell us next year in Boston. In Boston next year, we'll expect an answer from you.
I think it's the mark of a true computational chemist when a Tautomer example is cute. Do we have some other questions for Yvonne? Well, once again, let's thank Yvonne